Okay, I think we're recording. So, oh, okay. Can I really not? Why? It's let me do it before. Okay. I'm just gonna move us way down here. Oh my gosh. Okay. This is John's computer, so I don't know what I'm doing. All right. So, sorry, that's gonna be in the way. I'll ask John to fix it when he gets here. Um, but we are going over topic 3. Give me a thumbs up, thumbs in the middle, thumbs down. How do you guys feel about this one? Okay, we got a mix. So if I'm going too fast, tell me. But I'm gonna try to get through it so we can take some problem set questions at the end. So if there are any that you guys want to look at, like have them ready. I can stay for a little while after, not too long. So let's get started on that. All right, so protein trafficking. Basically, the whole idea of protein trafficking is we have our proteins being translated from mRNA on the ribosomes, which live in the cytosol, right? But we need our proteins to be in the nucleus or the mitochondria or the chloroplast for plants. Like we need them to be localized in different places around the cell. An example of that would be like DNA polymerase. DNA polymerase is a protein that we need for DNA replication. It's synthesized on the ribosome in the cytosol, but we need to be able to get it back into the nucleus. So we have a system using amino acid sequences called signal sequences to get proteins where they need to be. And those amino acid signal sequences are basically binding sites for carrier proteins that will bring them to whatever organelle that they need to be at. So central dogma, reminder of that, basically just the idea that DNA is transcribed into mRNA, is translated by ribosomes into proteins. And that is what happens in the cytosol. And then from there, we can kind of take 2 diverging paths. So one of them is going to like whatever organelle, if you're going to stay in the cytosol or go to the nucleus or the mitochondria, whatever. And then we have a whole network of organelles that are kind of interconnected called the endomembrane system, which we access through the endoplasmic reticulum. So any protein that needs to be anywhere in the endomembrane system has to get there through the ER. Yeah, for sure. And these slides will also be sent out, and we will upload the recording. So I am gonna move on now. These will be available after. So basically we start off with our free ribosome, right? And then if we are moving something into the endomembrane system, into, the endoplasmic reticulum, or anywhere else, the Golgi bodies, the lysosome, or to be excreted out of the cell, we have co-translational transport. So does anybody remember what that is? Wants to like take a shot at a definition? It's okay if not. Yeah, go ahead. Exactly, yeah. So what we want to do when we're putting something into the ER is we want, as our little signal sequence comes off of the ribosome, to have it bound and brought into the ER or over to the ER as it's still being translated. And we will talk about why that is after, but the term for that, I would know it. If you think you're going to forget it, maybe write it down on your exam note sheet. That is co-translational transport, and that is what happens to get something into the ER or into the endomembrane system. And so when we have our co-translational transport, that brings our ribosome onto the surface of the ER. And so when we talk about the rough ER versus the smooth ER, the rough ER is studded with those ribosomes that are doing co-translational transport. And so from there, we should technically have ER like here, but that's okay. So we have vesicles that will transport to the Golgi body, or from there the lysosome, the plasma membrane. And what a vesicle is it's kind of just like a shipping package. It's made of lipids. It is stable in the solvent in the cell to transport proteins between organelles. So endomembrane proteins, once they are in the endomembrane system, cannot come back out. The only time that we would see them potentially coming back out is during UPR, unfolded protein response. And that is just to protect the cell from, we don't like unfolded proteins, right? They can aggregate, they take up space, they don't function. So we want to get rid of them, and we need a system, a highly regulated system, to take unfolded proteins back out of the endomembrane system. But we need chaperones to do that. And a lot of that involves like kind of disassembling the protein to get it back out of the pores in the ER, which are really, really small, and a folded protein couldn't do that. So a folded, functional, stable protein is not going to be able to come back out. It actually has to be folded inside of the ER because the pores are too small to fit a folded protein through. And then once we're in the endomembrane system, we have a couple of outcomes. We can have a retention sequence, which means if you have like an ER retention sequence, then you will stay in the ER. You can have a Golgi retention sequence, but otherwise it's kind of like one of those moving sidewalks at the airport, where it just kind of moves you all the way out. And then if you don't have any retention signal sequence, you will be moved straight out of the endomembrane system and secreted into the outside space, the extracellular area. But some examples of that are like hormones, which would be secreted for signaling at long distances, or transmembrane proteins, which we're going to bring just into the plasma membrane but aren't going to leave, or catalytic enzymes, which are used to break certain things down in the cell in the lysosome. So the reason that we would not put, or actually, does anybody know the reason that we wouldn't put an unfolded protein in the lysosome, or do you know where it goes instead?
I think it was too big to fit into the reason why some of them were for a little bit was because of had issues with the pathways.
I mean, kind of. It has a more specialized place that we go when we want to degrade proteins. So proteins will go to the proteasome, which is kind of just like a garbage disposal specifically for proteins. I'm guessing the reason for that is the enzymes that are there are more specialized for protein degradation. But when we have our UPR and our chaperones bringing an unfolded protein back out of the endomembrane system, we are going to bring it to the proteasome. So far, so good. All right, so this is just a visual representation of that. We have our post-translational transport. So these are like the enzyme or organelles. Oh my gosh. Organelles that we will go to once the protein is fully folded, which means some way our folded protein can get into these, especially for the nucleus. We even see in this illustration, it has huge pores. So we can translate our full protein and then bring it to those organelles. And then we have co-translational transport, and that's going to be the ER and the endomembrane system. A lot of these you don't have to worry about. We don't really talk about peroxisomes or chloroplasts, but the rest of it, kind of be familiar with. I like this graphic because it's a good way to see how, they end up going to different places and how they end up there. But co-translational transport, just remember that one for ER. And then this is a little bit of the difference between like the kind of translation that we're going to have for a post-translational protein and a co-translational protein. So for post-translational, we have our mRNA translated by the ribosome into a polypeptide that is folded in the cytosol, and then it's brought to its ultimate location. Where for our co-translational, this is one of the pores or the translocation channels in the ER, and it's really, really small. So to be able to fit a protein in there, the ribosome has to dock, and this is where on the rough ER you would have like that studding. And the peptide, as it comes out, is threaded through this translocation channel, and then it folds inside. So this is why when we have a folded protein, we can't get it back out. We would need a chaperone to like hold it in the right conformation or partially unfold it to bring it out to the proteasome. And this is just a different representation of that. Something that I liked about this one is that it shows a lot of vesicle transport between the different organelles. So there is like no physical bridge or pathway between like the ER and the Golgi, or the Golgi and the lysosome. but within this system, we're going to kind of be using vesicles to transport from organelle to organelle. Yeah, and then this, oh, something that I wanted to highlight here is basically once we have a protein in the cytosol, like it has some options, it has some mobility, it can go depending on its signal sequences to a lot of different organelles. Chloroplasts, mitochondria, once it's in there, that's kind of like the endomembrane system where it doesn't really come back out. And the nucleus, we will talk about the whole import and export cycle. Proteins can move in and out really easily, but only if it has both a localization sequence and an export sequence to be brought back out. So, signal sequences. I think Laura calls them a zip code. I don't know if she's called them that this semester. Huh? Yeah, I mean, she calls them signal sequences because that's like the official name, but I think she uses zip code as an analogy. So it is the sequence of amino acids that kind of chemically tells other proteins where it belongs, where its final home is going to be in the cell. And it is a part of the primary sequence of amino acids. It is translated from the mRNA. It's not a post-translational modification. It's not something that's added after to tell a protein to pick it up. It is a part of the primary sequence. This is not my computer. I was afraid of this happening. Okay, we'll see how far we get. I think John is going to be here soon. I'm actually going to text him so fast. Sorry. My computer doesn't work, so I can't do Zoom from my computer. Your computer is dying. Lol. Okay. So, yeah. We have trafficking proteins that will bind to our signal sequences. And it's because they have like complementary chemistry. So, you know, maybe we have a really hydrophobic signal sequence like we do in the case of the ER. That means that the SRP, which picks up the ER signal sequence, probably also is hydrophobic. Or maybe we have a lot of positively charged amino acids on our signal sequence. Then the carrier trafficking protein that picks it up probably has a lot of negatively charged residues. So we are making this interaction through affinity between the signal sequence and its binding partner, the trafficking protein. And we already talked about this. We have ER localization, and then we also have retention signals for kind of anywhere past the endomembrane system. We don't need those for any other organelle because they're just like their own isolated place. So it's not like you're going to be shuffled to the next organelle or out of the cell. So co-translational transport and retention signal sequences are only applicable to the endomembrane system. Okay. And this is where we talk a little bit about like the signal sequence chemistry. I did not understand what this diagram was trying to tell me at all when I took the class, but basically, this NH3 plus, this is showing that this is an N-terminal signal sequence. So it's at the very beginning of the polypeptide. This COO minus is telling you that it's at the C-terminal. So our ER retention signal sequence is at the very C-terminal, like last end of our protein, and then like these would be in the middle of the protein because we don't see either terminus. And then we have our different like positive, negative. Yeah, some of the things that we use in the slides come from Laura's videos. And then we have our hydrophobic ones. And the most hydrophobic one, the one that like I would kind of be familiar with, is the ER signal sequence. So let's see if I have it on the next slide. Yes. Oh yeah, you could do that. I feel like these are pretty distinct though, so that might look too close to the pink. Okay, so yeah, our ER localization, it is a super hydrophobic signal sequence. So that means as it's being translated by the ribosome and it comes off at the terminal end, we want something super fast to protect it from the cytosol because the cytosol is aqueous, it is water-based. So our SRP, our signal recognition particle that probably has like a hydrophobic binding interface, is going to come super quickly. Hi, your computer's about to die.
It is.
Yeah. Yeah. So give us 1 second to plug this in, actually.
Mhm.
I was just said, give us a second to plug this in. Just in time, thank you.
Hi guys, enjoy.
I don't know where. Here, you put it. I don't know where it is. Okay, yeah. Sorry about that. So the SRP is going to grab our ER localization sequence, and then it's going to bring it over to the. It's going to bring it to the rough ER, and it's going to dock, and then yeah. We thread it through that pore. Once we're inside, also we have endonucleases that will cleave off that signal sequence, and then there are chaperones in the ER that will fold that ER protein. And like I said before, like if we don't have that retention sequence, it will be pushed through the endomembrane system and secreted out of the cell. Mitochondrial is similar. Mitochondrial pores are also small, but it doesn't use co-translational transport, which I don't have an answer for why that is. But for our nuclear localization sequence, this one is a little bit different in the sense that our NLS and our NES sit on the surface, the outside surface of our folded protein. So anytime that we need to send a protein to the nucleus, we need it to be fully folded before the NLS can bind. So there are some questions on your problem set about like what would happen if you had an ER signal sequence and a nuclear localization signal sequence. Basically, the trick to these is like for the NLS to be bound by important and brought into the nucleus, we need a fully folded protein. And if we have something, you know, coming off before, like an ER localization signal sequence, that is going to grab that region, that signal sequence, before the protein is done translating, before it can be folded in the cytosol. So it just won't have the opportunity to fold and to be bound by important and to be brought into the nucleus. Even if we have something on the C terminal, like if that's still coming off and then is bound, that doesn't give the protein enough time to fold and be brought into the nucleus. So you kind of just don't really do nuclear transport if you have another signal sequence. Does that got for mitochondria? No, so neither of these need retention sequences because neither of them are like trying to push you to other organelles or to out of the cell. What we will talk about with the nucleus is that if you want to be able to go in and out, you need a localization sequence and an export sequence. And a lot of nuclear proteins will have those, right? Because during mitosis, we will degrade our nuclear envelope. So things have to go back in, they have to come out. We see a lot of that cycling through. But they don't need a retention sequence because they're not going to be like pushed out anywhere. Okay. Yeah, so we talked about this in class. I think this is one of your clicker questions, but some proteins don't have a signal sequence. And in that case, there's just no binding site for a trafficking protein to come and grab onto it. So it won't be brought anywhere. It will just stay where it's translated, and the ribosomes live in the cytosol, so it will stay in the cytosol. And that's fine. That's not like a defect. It doesn't mean that it's missing its localization sequence. Could mean that if there was a mutation, but a lot of proteins just do need to work in the cytosol. My favorite example is kinesin. We have ones that like will walk along our spindles. Like there are just tons of proteins basically everywhere in the cell. So, yeah, nuclear proteins we just talked about need to be fully folded. The pores are really, really, really big compared to like the ER. They're huge. So to get them in and to allow the NLS to bind, we just need a folded protein. And ER and mitochondrial location sequences are cleaved, which we talked about. So this is just a diagram of that. I will send this out. I wouldn't worry too much about this, but if you're a visual person, it might help to like see the different pathways where our proteins can end up. And this is a better picture of ER transport. So we have our ribosome, our small and large subunits, and our mRNA being translated. And then, yeah, this is our signal sequence, and it's bound by SRP and then brought over and threaded through, and then it folds inside. And we have chaperones that will help it fold, and we'll have nucleases that will cleave off that signal sequence. So if, just hypothetically, we could export a protein from the ER, would we be able to get it back in? I see people, yeah, shaking their heads. Does somebody want to say why? Somebody knows. Go ahead. Right, exactly. So it's not like you need a signal sequence every time you're trying to get into an organelle. It's not like you've been there once and you can kind of just like bind again and get in. After it's cleaved, and we need that. It's kind of like a key. Okay. I actually changed these slides and I forgot to like refresh your computers. I'm like, why aren't these how I expected them to be? Anyway, no, it's fine. All right, so quick practice question. Transmembrane proteins have an ER localization signal sequence, and then they also have a plasma membrane retention sequence. So as they're like being brought over to the plasma membrane in that vesicle, they don't just get pushed through, they'll stick in the membrane. So where would you find the membrane receptor protein if you engineered it such that it no longer had an ER signal sequence? Assuming that it doesn't misfold or get degraded. Would it be secreted outside the cell? Would it stay in the ER? Do you know? It's okay if you don't want to answer. Yeah. Yeah, exactly. So this is the kind of question, I know this is kind of a reiteration of stuff that we've already talked about, but this is the kind of question that you might see on an exam. Just because you have your first one coming up, like as a heads up, Laura really likes to ask this style of question where it's like, okay, well, I know that you know or have it written on your sheet like how this works normally, but what would happen if you broke this part of it, like took out this localization sequence, if you had a loss of function mutation, whatever. So kind of practice that, be ready like when we're looking at how things work to also understand how they would be affected like if we broke something so it didn't work. That's a good style question to practice for the exam. And that's just the answer to that. Okay, so just an example of like moving things through the endomembrane system to be secreted. We have hormones. Hormones are encoded in the genome. A lot of them are proteins, and because they do signaling at a distance with other cells and other parts of the body, they need to be secreted. So we have our ribosome here, right? It docks on the rough ER. It threads the peptide through the translocation channel. We have chaperones that will fold our peptide into a globular protein on the inside of the ER. And then we have our little vesicles that kind of like bubble and bud off of the organelles, because these organelles all have fatty membranes. So it's kind of like a lipid micelle, if you guys talked about that, like in your Gen Chem, that will bud and then fuse with the next organelle. So we go through the Golgi complex and we have processing that's happening as that's going on. But then, you know, after our processing, after we've made it through the Golgi complex, our vesicle will basically fuse with the plasma membrane of the cell because they're made out of the same material. It's kind of like the like dissolves like thing where they will be compatible and it will just kind of fuse into the plasma membrane, and then whatever's in it as it fuses gets pushed onto the outside of the cell. Go ahead.
Well, I don't think I've ever remembered the new session starts.
On Tuesday, which I think is the 3rd, there will be an exam 1 review session. So yeah, we will go over all 3 of the first topics and answer questions like, you know, about note sheets, problem sets, exam tips. So yes. 7 p.m. Yeah. And we will send out an email for that too. Not me, but okay. And this is another kind of example to illustrate that. Lysosome, we kind of use it to break down things that we don't want in the cell, waste, mostly not proteins because those will go to the proteasome, but the lysosome is actually a part of the endomembrane system. So if we have an enzyme, which enzymes are usually proteins, they can be RNA, but enzymes are usually proteins. So if we have a enzyme that we need to bring into the lysosome, it needs what does it need? It needs 2 things. I know. Anybody? Somebody give it first, yeah. Yes, exactly. So we need ER localization to get it there, and then lysosomal retention to keep it there. Because if we don't have a retention for anywhere in the endomembrane system, we will be secreted out of the cell. So yeah, that I think is it for sorting, trafficking, signal sequences. Do you guys have questions on that? I feel like that one is pretty straightforward, so I will give it to John.
You're recording, right?
Yeah.
Okay, good.
I hope.
I hope
I
too.
hit record.
Okay.
I don't know.
Hi guys, I'm John. No, I'm kidding. Okay, so we're gonna move on to the GTPA superfamily. So on the corner right there, I know the projection isn't like awesome, but on your slides it's gonna look better. It's just like a little visual of the different proteins present in the RAS family of G proteins. These are all GTPases. If you ever see the little ASE at the end of a word, it's genuinely speaking an enzyme that helps break down the name of the enzyme. So like a DNase breaks down DNA type of thing. So GTPase breaks down the phosphate bond between one of the 2 phosphate groups of GTP. And so RAN, specifically the one we're talking about, helps to regulate nuclear import and export, which are the cycles we will go over in just a second. So again, RAN is a GTPase, but very important here, it could do its job by itself. It just sucks at it. Takes absolutely forever. So it needs little helper proteins. And again, RAN related to RAS. We go over RAS and other topics as well. But yeah, so that's just the superfamily. There's a visual at the corner if you really are into visuals. All right, gaps and gaps. Gaps and gaps are the proteins that help the regulation of the RAS family's proteins. So we have GEFs first, guanine or guanosine nucleotide exchange factor. Guanine isn't actually the name of the nucleotide, but guanosine if you're really like into nerdy stuff like that. So it just helps the exchange of GDP for GTP on the active site, because it causes a conformational change, which aids the protein that it's being used on to let go of GDP. And so it decreases the affinity that GTPase has for GDP for GTP. It's not like it's just like, oh, I'm here, oh, let go. It's like there's a whole like process of things that happens from binding to conformational change to diffusion. And so this is to facilitate the binding of GTP. And so ultimately what you want to do is you want to get rid of your GDP, get your GTP in. And then we have GAPs, which are GTPase activating proteins. Laura clarified this. I'm going to clarify it, write it down, make a note, tattoo it if you want. GAPs are not enzymes. We are not writing GAPs are enzymes on the exam because we do not want to be wrong. Okay. Again, GAPs are not enzymes. And so they can hydrolyze GTP, or I mean, sorry, the GTPase, which in this case is RAS, can hydrolyze the GDP to GTP or the GTP to GDP on its own, like I said, it takes absolutely for, yes.
Does that have gas? Gas also not enzymes, but gas
Neither of them are enzymes, right? Yeah, neither of them are enzymes. It's just GAP is more commonly said as an enzyme. It's like the mistake that occurs the most. We do not want to make that mistake. Yeah, as I think what gets lost is that, oh, like activating and then it like sounds like enzymatic activity. It's just not. So like I was talking about, rats can get the hydrolysis process going. It just takes absolutely forever. It sucks at it. So you need the little GAP protein to come in and help facilitate that. And so what we're gonna do is we're gonna get the GTPase bound to GDP at the end of the day. And so to clarify it, it is not an enzyme. And it just activates the GTPase's intrinsic capabilities. And that's why it is so vital. Because this is a very important process as we're going to look at the import and export cycles on the next slides. But it needs to be done in a timely manner so that the procedures are optimal time-wise. Yes? I don't know what that line is supposed to mean. It's the
Okay.
second and
Yes,
last
when
line.
we talk about, we're going about this, right?
The second to last.
Yes, that one. Yeah. So when we talk about import export, we are gonna talk about like where gets and gaps are found respectively. That is on the diagram, we will go over that. And then it shouldn't say that it makes it active, but it does accelerate its access. So it makes the GTPase activity trans faster. Does that make sense? I thought it was the gap, that's the kind of facilitating.
Yeah, that is specifically for the import and export cycles. So we're gonna talk about that soon. But in general, the gap is just facilitating / activating the enzyme's intrinsic capability. That line is specifically for the import and export cycles that we're about to go over. So it will make more sense like in a second. Huh? Oh, okay. Okay, so the import cycle, if you're a visual learner, there's the diagram. Do they get the diagram on the exam?
Yeah, you guys on the exam, you get the amino acids and you get the import export cycle.
Okay, so if you're a visual learner and you like the arrow system, there's the arrow system for you. I'm more of like I need to read it like 12 different times till I understand the type of person, so I like the written down version better. But the visuals do help, I will not lie. So first we are starting on the cytosol at the bottom right. And so important is gonna come in. It's gonna be free and just roaming around the cytosol. And it's gonna bind a cargo protein that has a nuclear localization sequence. John, why is that important? That is important because if you don't have the NLS, important can't find it. Important can't bind it, can't be brought into the nucleus. So upon binding, this complex is gonna enter the pore. So right there on step number 2. And then step number 3 is gonna be inside of the nucleus already. And so RAN is gonna come in and it's gonna be it's gonna go from its GDP bound state to its GTP bound state with the help of RAN-GEF. And so GTP is gonna be activating it. And upon activation, RAN is gonna go in there and it's gonna cause a conformational change on the cargo / important complex. Why is that important, John? Because once that happens, the conformational change is going to cause a change in affinity between the two proteins, and they are going to diffuse from each other. So the cargo protein is now going to be available in the nucleus to perform its function, and important is going to be bound to RAN. John, why is that important? Okay, important needs to be bound to RAN in order to leave the nucleus.
John, why is that important?
Huh?
John, why is that important?
Don't worry about it. Okay, so once we are in the RAN bound to the important step, it is going to go through the nuclear pore again, going out this time, back into the cytosol. And in the cytosol, the last step is we're going to get a RAN gap to cause hydrolysis of the GTP bound state RAN, which will then cause a conformational change on itself, letting go of important. John, what happens if we don't have access to a gap in the cytosol? We're not going to be able to get important free. We're not going to get the products that need to go into the nucleus, and we are going to have a high concentration of important that is unusable in the cytosol. John, what happens if you don't have GEF available in the nucleus? Which is what the last thing was referring to. GEF exists in the nucleus for this reason. If you don't have GEF available, then you won't be able to get RAN onto its GTP conformation, which then you won't be able to let go of your cargo with important. So that is why the presence of the activating and exchange factor proteins is vital on this cycle. So again, GEF lives in the nucleus, GAP lives in the cytosol. Good? Good questions? You understand now? Better, at least? We got rid of that on the slides, so like the updated slides won't have that. I agree, that is very confusing.
Yeah, that was the old unedited version of the file. I didn't make it.
So- When RAN is GTP, there's like a switch in important to let go of the protein.
So it binds to important causing a conformation.
Oh, okay.
Yeah.
Do you know how like how RAN gets back
Into the nucleus? I don't think you need to focus on that now. Do RAN going back into the nucleus? Yeah, I don't think you should worry about that. Anybody else? No? Okay, we're gonna go on to the export cycle. Okay, export, it's kind of like reverse. But we're gonna have a main difference here, which is later on. So we start in the nucleus this time around because export, we're trying to get rid of the stuff that we need to get rid of from the nucleus out of the nucleus. So with the help of a GEF here, GEF lives in the nucleus, GAP lives in the cytosol. With the help of a GEF, the GDP is going to dissociate from RAN, which will then be able to bind onto a GTP. That's the first step. Step 2, you're going to get a cargo protein, and it's going to contain NES. What's an NES? Not the video game.
I know.
Yeah, I know. I know. What's an NES? There we go. Okay. So protein's going to have the NES, right? So then we're going to get RAN GTP. That's going to bind to exportin. That happens, exportin is going to get a conformational change. With the conformational change, it'll be able to bind the cargo protein. Then we have a RAN GTP bound to exportin, which is bound to its cargo, which is the step number 2 right there. Step number 3, they're going to go through the pore. And then that leads on to step number 4. So then with the help of a GAP, we're going to get hydrolysis of GTP into GDP, which is going to cause a conformational shift, which will lead to our exportin dissociating away from RAN, and then an inorganic phosphate being released from the GTP onward. And so it's a RAN GDP, an inorganic phosphate, the cargo protein in the cytosol, and exportin becomes free. So all that happens in that one step. And then upon that happening, exportin is now free to just go back into the nucleus. So then it just diffuses through the pore again. Yeah. No, I think that just more is produced and then the concentration of RAN is just like different on the cytosol and the nucleus.
So outside of nucleus, the grocery is GTP.
Yeah, it gets hydrolyzed. And then once it's hydrolyzed, it causes conformation of change in RAN, let's go exported, which causes a conformation of change that was stored.
So I guess it would be worth to know that just import and export both get reused essentially.
Yes,
So they cycle back.
they're very important, which is why Laura loves asking the type of question of like, oh, if I have a faulty gas in the media, what would happen and what would that cause to the concentrations of whatever that you want. All right, anybody else questions? No, okay. So transcriptional regulation, we're gonna go over the different steps that could be used as transcriptional regulation. So transcription factors is what regulates RNA polymerase binding to the promoter region of different DNAs, or I mean different genes in the DNA. So no transcription factors, polymerase is not going to be able to bind. This RNA polymerase is different from DNA polymerase. Why, John? Because RNA polymerase uses uracil, DNA polymerases do not use uracil, they use thymine. So regulatory transcription factors are very gene specific. The cell can activate different transcription factors depending on what types of proteins it needs to make and what different proteins it needs to use, and cell stress responses happen, and transcription factors are like very cool to learn. And so there's positive and negative regulations that occur through regulators. And so positive enhanced transcription and negative suppressed transcription. This is important because if naturally you're expressing a protein, all of a sudden you have too much of it, you need to stop producing it. So you stop wasting cell resources. I couldn't figure that word. So then continuation, that is chromatin. You don't need to know the two different names for the different types of chromatin. All you need to know is that chromatin is DNA wrapped around a molecule or a protein called a histone. And on the closed state of histones, it is hard to access the specific genes you're trying to target. And then on the open conformation, it is easier, which is how the polymerase and the cell machinery go in and start transcribing. If you really want to know, the open state is called euchromatin and the closed state is called heterochromatin. You don't need to know this for this class, I don't think. No. Anyways, yeah, go ahead.
So I just thought it was interesting. Basically, we're interested well. Heterochromatin remodeling, which is a kind of transcriptional regulation, can be passed epigenetically, which means that like the way that your genes are regulated and basically the way that your DNA is wrapped around the histones to silence some expression can be transmitted to future generations. So like
I agree. Anyways. All right, moving along. RNA modification and processing. So RNA has to be processed and made stable into mature RNA. You have different enzymes in the cytosol that are looking for inactive mRNAs to destroy. It's just a process that happens. Just regulatory, it prevents a whole bunch of different problems in the cell. So how are they stabilized? So if you look at this fine drawing that some fine person on the internet did, you have a Pac-Man trying to attack the 5' end of the mRNA, and you have a different one trying to attack 3' end of the DNA. 5', we're gonna have a cap. So it's covalently added. Very important to know that it's covalently added if you get asked. I don't know if you're gonna get asked, but if you are, you can. Let me give my spiel. If you get asked, they are covalently placed. And so it prevents RNA from being chewed by the nucleases that are looking for it in the cytosol right there. And it has a specialized guanine that has slightly different structure, and it's added in the opposite direction, which is why it doesn't allow the nuclease to find the strand. And then on the 3' end, you're going to have a poly A tail. And so it's a long sequence of adenines, which is why it's called a poly A. and it's necessary for export. And so eventually the mRNA can get degraded, because the adenines diffuse away. But that's like a time-consuming process. It doesn't usually happen. They're both there in order to protect the mRNA from nucleases from destroying it. Any questions? You want to add anything? Okay. Continuation, RNA splicing. Very important, guys. So if we look at that drawing of the DNA, you're gonna have a premature mRNA. mRNA stands for messenger RNA. Premature mRNA stands for premature messenger RNA. What is this? This is the raw RNA that gets transcribed from the polymerase that needs to be quote unquote edited to get only the coding portions together. You do not need introns for your protein to be translated. And so to get rid of those introns, a process of splicing happens. So introns are the areas that will not be translated. So it's why we get rid of them. It's just like they're just there. And so the enzyme that's going to go in there and splice the RNA to make it a mature messenger RNA is going to be the spliceosome. And so there is a process, a convenient process. Wait, hold on. Yeah, right here. So there's a convenient process that goes on in order for you to have different variations of the same protein called alternative splicing. So it is not because I have all of those exons that I necessarily need to use those exons for my protein. I can cut out specific parts of the entire mRNA to get different looking proteins. And so right here, we have example A, where you get exons 1, 2, and 3 on the final protein, and then, or on the final mRNA. And then on the second example, protein B, you have exons 1, 2, and 4. So what happened? On the first one, we got rid of exon 4. On the second one, we got rid of exon 3. This is the same gene, guys. It's just you're getting the different exons in to make the protein look different. Good? All right. So right here, the final step, I think there's one more slide after this, right? So stability of mRNA. So an mRNA's lifespan can be, lifespan can be regulated. And this involves RNA interference. This is something we'll talk about later in the course, so you don't have to worry about it. You just have to know it exists. And so cells can control the overall degradation rates of mRNA. And this is important because, like we were talking about, production of different proteins can be necessary at different rates at different times. So it's just another way for the cell to prevent bad things from happening. And now we have translational control, which is how we modify, or we modify proteins before they're actually translated. And so right here we have the ribosome binding accessibility, and different proteins can block the ribosome's ability to bind messenger RNA. We will go over some specific examples later on in the course, right? Yeah. And another type of translational control is mRNA localization. So where specifically the mRNA is located can affect the expression of the protein or the gene, sorry. And so these are just questions, or like rhetoric questions. I don't know. I don't like explaining it like this. It just basically means that depending on where the mRNA is located on the cell, different things can affect the rate that it will be translated. Yeah. So right here is just a convenient little chart about the different types of translational and transcriptional control in different types of cells. So for prokaryote cells, or prokaryotic cells, they don't have histones specifically. They do have histone-like proteins that they use to store DNA a little bit more efficiently, but you do not need to talk about that. So they don't package the DNA into chromatin, which is why they're single cell. They don't have. So they don't have a nucleus, so they don't have RNA processing because it only happens in the nucleus. What does happen though is simultaneous translation upon transcription of the DNA, or yeah, of the RNA. So what's happening is they're gonna have RNA polymerases go in, and they're gonna transcribe the RNA. At the same time, they're gonna get a ribosome and ribosomal machinery in there, and they're gonna be translating proteins. They do process the RNA. We don't need to talk about it. And then right here, we have the eukaryotes that do package the DNA into chromatin. If you guys don't know, chromosomes are just a lot of chromatin together, which is how they pack onto that like specific format. And so eukaryotes have nuclei. So the mRNA is processed and it is transported. And this is a big difference right here. I think this is the most important part out of this entire table. Eukaryotes regulate transcription with transcription factors called basal transcription factors, while prokaryotes use transcription factors called sigma factors. Okay, and so here is just the chart is just a way for you to have these differences and similarities for both. If you are looking to write that down, you can compress this and make it more like space efficient for your note sheet for your exam. But they both regulate mRNA degradation rates. They both regulate mRNA localization. They both regulate rate translation, and they both regulate protein activity and degradation. So they both.
No, so
Eukaryotes have basal.
okay,
Prokaryotes. Okay. So to clarify, eukaryotes have basal transcription factors, prokaryotes have sigma factors. Okay, so this is just like a step-by-step visual, if you're a visual learner, type of flow chart for the different processes of gene expression regulation, and we've covered all of those. This is also a convenient way of packing a lot of information onto like a smaller space for your note sheet. So you just have the different kinds, ranging from having the DNA exposed to the transcription machinery all the way until you have protein expression, or I mean, protein translation and degradation. Yeah.
For the exam, so you know, guys, I would know like at least one example of each of these.
Yeah, when we
there
took,
were questions for like, oh, you know, give 3 levels of gene expression regulation. example of each. So,
yeah, and I think, correct me if I'm wrong, but that was one of the questions on the problem set too.
oh yeah.
Yeah, so just know an example of each. There are multiple for each, just go with the easier ones. So, okay, so that's it for, oh no. So that's the eukaryotic. There's a difference between the prokaryotic, or a couple of differences. Because the prokaryotes don't compress their cell or their DNA into chromatin, and RNA processing for prokaryotes is completely different. That's just out of the window. We don't talk about it. And because there's no nuclei in prokaryotes, you just don't need mRNA transport because it doesn't need to be in the cytosol. I mean, it needs to be in the cytosol. It doesn't need to be out of the nucleus. All right, and that's it. We're gonna hang around a little bit for questions. But that's gonna be it for the recording. Any last questions about the last couple slides? No? Alrighty.
I have a comment.
Oh.
Just so you guys know, it might be good to start looking at your note sheet. I don't know if Laura has told you this yet, but you know that you get a full piece of paper front and back.
She did. She
yeah,
said
I
that in
know
class.
But on your final exam, you get to bring all of your midterm note sheets and then like a new final exam note sheet. So actually like put some thought into what you want to be on it. Don't just like, you know, do it in, you know, a half an hour before the exam. And you do get to bring it in.
I've never done that before.
It's double-sided on like a full.
Double-sided, yep. Yeah, it's pretty.